So hello, everyone. My name is J.Sepulcre. I'm a faculty member at the Gordon Center for Medical Imaging at Mass General Hospital. And my lab works on system neuroscience and neurodegeneration research. I'd like to thank Shumin and the organizers for inviting me to share this work. And it's really a great pleasure to participate here and to talk about molecular imaging and brain connectomics. There are two key developments that have produced kind of a revolution in recent Alzheimer's disease research. One relates to PET imaging, right, the development of new tracers. And the other one relates to the advances on brain connectomics, right, the study of cerebral networks in the context of neurodegeneration. So, PET imaging has allowed us to more precisely kind of characterize the hallmarks of the disease, right, amyloid and tau accumulation in vivo, that's the key. And at the same time, imaging technologies or techniques such as, tractography, diffusion imaging, or, functional connectivity have given us the ability to understand how large-scale brain networks really break down, right, during the process of the disease. So in my field, we support that, AD as well as PD or ALS are all kind of network diseases in a very profound sense. In my lab, we like to capture this idea with this well-known sentence. Not only, do neurons that fire together, wire together, but also neurons that, that wire together really die together. And in the middle of this revolution, there remain some, great challenges to really integrate the PET data, right, into the network nature of Alzheimer's disease, right? Challenges to integrate kind of the key molecular biomarkers like tau and amyloid on its kind of connectivity identity. That's why in my lab, we have devoted most of our efforts to do this type of integration, right? Obviously, to ultimate the goal of, with the ultimate goal of kind of elucidating the relationship between network degeneration and its underlying biology. For many decades, Alzheimer's disease has been conceptualized to as a kind of a hierarchical progression from medial temporal lobe, right, to other regions of the cortex, like frontal and parietal cortex, like in the Bragg stages. This is like a kind of a progressive building up of pathology, right, from temporal to the temporal cortex. But until very recently, we didn't really have the proof of that in vivo. We didn't have the kind of the evidence to about the regional dependencies in the accumulation of tau and amyloid. So in this initial study that I'm showing here, we were kind of the first to start evaluating those spatial dependencies across brain areas in aging population. On the left is just the typical maps of based on PET signal intensity, but on the right, we implemented a hierarchical clustering approach that shows us a very detailed, hierarchical structure, between these specific regions in the cerebral cortex, particularly for tau, right? The tau accumulation pattern here in red and one cluster in red, one other cluster in green, they correspond to this type of spatial dependencies between the accumulation. And we also confirmed the existence of these regional dependencies between amyloid-related regions. For instance, in this medial parietal area, shown here in red, in which there are green areas locally surrounding it, right? So we basically observed in the study that it seems that there are areas that work together. Somehow they coordinate together in this process of the accumulation of tau and amyloid. But critically, in this study, we also observed that tau and amyloid don't work in isolation. Rather they display very specific interactions in their building up, right? If we focus our attention in this little matrix here, we can observe that not only high modularity between tau and tau interactions or between A-beta and A-beta interactions, we also observe that there is a very, very specific and interesting intermodal interactions between tau and amyloid, right? Particularly in the context of, in the case of tau to amyloid in a specific parietal and frontal areas. And if we display the same findings in form of a graph, right? It's easy to see that tau and amyloid clusters behave differently. For instance, amyloid clusters are, kind of densely grouped. While in the case of tau clusters, we can see there is one tau cluster here in the middle that seems to be very essential to do the interlink, right, interface between these two pathologies. Well, these results we thought were very promising, right, regarding our aim to understand these spatial dependencies in the AD pathology. So we then began to wonder whether we could kind of push forward the investigation of tau and amyloid interactions at the network level, right, but with much more kind of high resolution. So if you do connectomics in your lab, particularly high resolution connectomics as we do, you probably have to deal with an enormous amount of information, right? For instance, this example is based on functional connectivity only from one single individual, right? And in this case, it's 10 million connections. So to deal with this huge kind of data and information and brain complexity, in my lab, we love to use graph theory. So graph theory is such a wonderful framework in our view, right, to deal with these high dimensional problems, because it allows us to transform this enormous amount of info into more tractable graphs and more, tractable features. As you probably know, graph theory was introduced in neuroimaging only 10, 15 years ago. And over those years, my lab has grown using and kind of developing these metrics, as you will see in the coming slides. Well, basically, graph theory is a branch of mathematics, right? It uses all these nodes and connections to form diagrams and network graphs. And it employs matrix algebra to calculate specific network properties, right? Network properties like centrality, segregation, integration, etc. And well, there are many types of graphs depending on the source of the data, the source of nodes and edges, right? So in neuroimaging, most of the times nodes are basically just gray matter voxels, while edges are more heterogeneous in nature because they can be based on, diffusion data, functional connectivity, and also PET imaging data, right? For instance, in this study, we use graph theory and centrality metrics, and we apply them to PET data to understand the network dimension of AD pathology at very high resolution, right? And here by centrality, I mean specific graph theory metrics that takes into account the weighted degree connectivity of all possible connections, right? And detects the hubs, right? The cortical hubs within in those brain networks. And by doing so, in these maps, right, we describe for the first time the cortical hubs of all these interactions between amyloid and amyloid voxels, right? Amyloid and tau voxels, tau interactions, and tau and amyloid interactions. And it turns out that tau and amyloid connectivity were extremely central, right? Not amyloid and amyloid or tau and tau, but tau and amyloid connectivity is really, really central. This is represented here with these orange nodes in the network or orange areas in the cortical surface here. So in this study, we found that tau deposits in these lateral kind of temporal areas links really well with tau and amyloid pathology across the entire brain. I mean, it couples kind of two pathologies at actually distant sites. So in our view, it seems that there are specific areas in the brain that might serve as a gateways or kind of an intersection point between the two hallmarks of the disease. So pushing forward that idea, of combining molecular imaging and connectomics in the context of Alzheimer's disease, well, more recently, we have also investigated the spreading patterns of tau and amyloid pathology in the human brain. First, we developed a graph theory approach to detect the cerebral pathway through which a tau and amyloid really progress, right? In this case, in this first step, based on cross-sectional predictions. Then we use longitudinal PET data really to confirm those predictions and to establish the actual directionality of the spreading, right? So it's important to highlight here that, the huge novelty is that because we were able to use really longitudinal data, right, time 1, time 2, of this PET amyloid and tau information, we can really assess and evaluate the real spreading, the real, and kind of the arrows of the directionality of the progression of the disease. Obviously, this is based on statistical approaches in which we take into account, right, the differential wave between voxels and time points. And with that, we obtain really interesting findings. For instance, this is an example of the tau spreading network using a temporal seed, right, in this blue area here. So this is the prediction. This is the kind of the confirmation of this longitudinal spreading. And we found that temporal tau, I mean, the tau accumulated here in this temporal area really is kind of, influence and spreads in time toward the anterior pole, right, toward the posterior parietal region, posterior temporal regions, or orbitofrontal cortex. And this is the case for amyloid, the spreading of amyloid from this PCC area, posterior cingulate cortex area, in which amyloid seems to spread more locally toward the posterior, right, and lateral parietal lobe. We obviously scale this approach up, to capture the main cortical hubs that tend to give pathology elsewhere in time. And we call this kind of the givers of signal, right? Or the receivers of signal. And we found that temporal areas tend to, for instance, in the case of tau, tend to give signal to really distant areas. While amyloid spreads more locally, again, here you can see it in this map, right? Even displaying a great deal of overlap between givers and receivers of signal. Interestingly, we applied this method to kind of evaluate the individual brains, right, to stage subjects at the individual level, and it works really, really well. And we also identified and discovered that there's some specific associations between the spreading patterns and the impairment in cognitive performances, particularly in tau spreading and in MMSE and memory. In the very last section of my presentation, I'm going to try to talk about something that is taking most of our time these days, and it's the integration between connectomics and genetics, right? So I think we kind of all agree that on one hand, AD is a network disease, and on the other hand, is kind of a disease that is strongly driven by genetic factors, obviously, right? So with this philosophy in mind, we're currently pushing forward the development of strategies to get the spatial similarity, right, between our imaging phenotypes and gene expression data, right, in order to kind of identify the neurobiological traits that can explain the vulnerability of specific brain networks. For instance, in our AD work, we took those spreading maps that I showed you before, and we compared them with the entire transcriptome, right, of 20,000 genes from the Allen Brain Atlas. And we saw that there are genes that display a very high spatial similarity with these, PET phenotypes of spreading, and not with the, kind of the PET conventional intensity maps. For instance, we observed genes like, MAPT and CLU in the case of amyloid, MAP in the case of tau and CLU in the case of amyloid. They match really well in these specifically kind of spreading patterns. And we also observed and discovered several gene sets that are related to kind of both types of spreading. And we did an interactomic analysis, and we saw, for instance, that in this common interactomic network, or this common background of genes that are related with both patterns of spreadings, for instance, ApoE and APP is really, really central in that network, which is quite striking. So to summarize, in this study, we went from PET imaging data, right? Tau and amyloid PET imaging data, to the identification of specific spreading patterns across several brain systems, and also to the finding of genetic traits that potentially can confer circuit vulnerability for the accumulation of tau and amyloid, or even for the accumulation of both, right, tau and amyloid. So to conclude, we think current strategies to integrate molecular imaging and connectomics have made important advances to understand brain diseases such as Alzheimer's disease. The connectomic dimension of molecular imaging, it is an ideal framework, in my view, to comprehend the spreading nature of tau and amyloid, and surely it can serve to also understand other neurogenetic diseases. And it looks like, today we are living exciting times, right? Because with the help of transcriptomic data that these resources available to us, we can close the gap between kind of brain complexity, pathology phenotypes and genetics. And that eventually will provide important clues to create network-based solutions and personalized treatments in brain disorders. And with that, I'd like to thank all the people at my lab that has been involved in these studies, particularly to Ibai, Sylvia, Ellie, and William. And thank you so much for your attention.